start recording now. I was about to forget. And Dr. Borish, feel free to share your screen and start your presentation.
Great. Thank you for that introduction, Ellen. Thank you for the invitation. Really happy to be here and happy to see several familiar faces and some, hopefully, that will become familiar faces over time. Yeah, so I understand that I have about 20 to 25 minutes, so I'm going to zoom through this talk. It's just like any talk, it's overly ambitious, and I won't get through all of it. So I've tried to put I think what is maybe the fodder for most discussion as it pertains to the journal article that I provided previously, but really that's meant to be sort of just part of a larger conversation about our work, where we're, as Val mentioned, we're really interested in how we can harness the endogenous plastic capacity of the human nervous system to increase recovery, primarily from stroke, but also in other neurologic disorders. So because I know I'm going to run out of time, the most important slide I'll put first, which is to acknowledge everyone who's working on this. Before we started, I was mentioning how fortunate I am to be in the environment at Emory and Georgia Tech, where I get the opportunity to work with just outstanding scientists, but maybe more importantly, outstanding collaborators who are highly interested in doing great science and enjoying it while they do. So I'm really fortunate to that, and I want to acknowledge also the members of the lab who are really doing all of the work, and I just get to brag about them. So that's fun for me. So just a really quick outline of what I'm hoping to, and I'm certain that I think we'll get through the first two bullet points, and perhaps time permitting, we'll just breeze through the last part, which is more of a forward thinking aspect of the talk. And also, if there are any, I know we're going to break out into discussion groups, but if there are any questions as we go or clarifications, I'm totally happy to be interrupted. Or if Val, maybe if you're monitoring the chat, if there's something that comes in that would be useful to interrupt me, that's no problem. I often don't take a breath when I'm speaking, so feel free to interrupt me mid-sentence, that's totally fine. So again, really the guiding question in our lab that we're really looking at is how can we actually measure and maybe more importantly, modulate neuroplasticity in the human nervous system. And we primarily use techniques that are non-invasive, and we are primarily looking at individuals post-stroke. However, we have really started to get very interested. I won't talk too much about that today, but really interested in how we might be able to augment the aging nervous system. And this is how we tend to conceptualize the sort of an organizing principle that really neuroplasticity is necessary for learning, and learning is necessary for function, and in the context of disease, the recovery of function. And the most potent driver of neuroplasticity is experience. So we still don't have, an effective drug that we can take to make, us more plastic, but experience is really what drives these changes in the nervous system that underlie memory formation, learning, and then again, in the context of disease, the recovery of function. So this is how we approach most of the questions that we ask in the lab and try to interpret what we see. And so there's a vast literature, and I don't have to tell this group that we know that the brain changes in response to experience. I always give this example to students in my classes, is that if you remember anything, if you remember that I have a bamboo plant from this talk, that suggests that you had some neuroplastic change in your brain where there is memory for that information that is stored. And so now your brain is essentially changed in some small way. And we can change the brain in terms of motor systems through experience training, as well as the restriction of experience, say in the examples of mobilization, or in the context of, say, stroke, in sort of learned non-use paradigm. But I think the important thing to know is that the capacity for plasticity is not abolished in the human nervous system across the lifespan or in the context of disease. So knowing that, we know that there is a potential substrate to exploit to try and maximize, again, function and recovery for these individuals post-stroke. And we know that stroke triggers changes not just at the site of the lesion and directly connected perilesional areas, but also remotely. And so we've known this for some years, and we showed this actually when I was doing a postdoc, is that we see changes in, say, white matter, indices of white matter structural integrity that are not just in the ipsilesional hemisphere, but are also in the contralesional hemisphere, as well as in regions of the corpus callosum. It's what's exciting, and I think is, what as a physical therapist by training is, there's really power in the fact that we as rehabilitation professionals can actually shape post-stroke neural activity to try and maximize this reorganization that occurs post-stroke. For those who are not stroke researchers, this is really the problem that we're trying to address. The idea here is that it's it's still the leading cause of long-term adult disability, and that depending on the estimate you read, the majority, what we can say confidently is the majority of individuals have persistent reduced ability to use their paretic arm, and that gets in the way of their ability to maintain their activities of daily living. And it's thought that this prevalence of stroke is going to continue to increase, and that we've gotten very good at acute management. So more people are surviving their stroke, which means that we have now more individuals who need rehabilitation services to try and reduce that magnitude of persistent motor dysfunction. So the question is, how do we potentially augment current gold standard therapies, which are still leaving the majority of individuals with persistent impairment? Of course, there's novel rehabilitation technologies that are coming out, whether it's robotics or virtual reality environments, an example here. What I'm just going to talk about today is neuromodulatory approaches, where we may try to augment endogenous plasticity through the use of these types of neuromodulation paradigms. And the one I'm going to focus on is the one that we specialize in, I think, in the lab, is non-invasive brain stimulation. And to add that to the conceptual model, really, we think that this is acting at the level, at a systems level, of plasticity induction. And so the idea here is that we know that individuals maintain the ability for experience-dependent plasticity across the lifespan, but it is diminished. So is there a way that we can augment to boost this endogenous plasticity capacity, to boost memory formation, which in turn has the potential to boost learning, and in the context of stroke recovery, boost that recovery of function? And again, we're using primarily TMS in our work. We have selected that mostly because we're interested in focal activation of cortical regions, but there are other ways and other non-invasive brain stimulation approaches like transcranial, direct, and alternating current stimulation paradigms that have the capacity to also modulate neural activity. But for the purposes of this talk, I'll just stick with TMS. And TMS, of course, can be used to investigate, again, plasticity, but it also can be used to induce plasticity depending on how the stimulation is delivered. So investigatory paradigms are usually single or paired, even in some cases triple pulse techniques, whereas neuromodulatory paradigms have some sort of repetitive component. So what I'm going to talk about is one way that we have combined TMS with peripheral nerve stimulation to try and induce plasticity in primary motor cortex. And so this has been demonstrated and known for over 20 years now, that if you repeatedly pair synchronous afferent arrival of sensory input, in this case median nerve stimulation of the arm, with TMS activation of the contralateral motor cortex, you will see an increase in the excitability in that region that was stimulated that persists past the period of stimulation. So that's what's shown here in B. Is the arrow is when PASS, this paired associative stimulation paradigm, was applied, and the time post PASS intervention. And you can see there's a persistent increase or elevation of excitability after the period of stimulation, but then returns to a baseline level 24 hours after the intervention. So what this has led the field to conclude is that we are inducing what we consider long-term potentiation or long-term depression like plasticity, where we see that at a systems level, these indirect indices, in this case, a motor evoked potential, a TMS evoked motor potential indexing cortical excitability changes with PASS, shows that this effect, this modulatory effect, evolves quickly. So you only need the stimulation to be delivered for 8 or 10 minutes, that it's reversible, it goes back to baseline, that it persists even after the stimulation has concluded, and it follows temporal rules. So this is important, that if you get synchronous arrival, you tend to potentiate activity in the region that's targeted. And if you have asynchronous arrival, say you have the median nerve stimulation arriving before delivery of the TMS pulse, you actually see the opposite effect. You see in a reduction or a depression like a phenomenon where you have reduced evoked responses. So that's the sort of the groundwork or basis for what we looked at. One of the open questions that we wanted to test is that this link between inducing plasticity exogenously with PASS, and does it actually show some influence or effect on motor learning. So if we think that neuroplasticity underlies learning, if we can induce plasticity, we would presume that we may induce learning. So I'll just jump right to the results. So we did a standard paired associative stimulation paradigm. The only difference here is that we also looked at the effective motor state. So in one case, we delivered pass while an individual maintained a low level, in this case, 15% max voluntary contraction during the delivery of this pass paradigm. So that's the pass active condition versus delivering pass at rest, where the target muscle, in this case, the APB, was at rest. And then the control condition was an active muscle contraction with only peripheral nerve stimulation and not paired with TMS. But TMS was delivered in sort of a sham paradigm. And then we looked at the effect on sequence-specific motor skill performance. And so what individuals would have a baseline assessment prior to pass, and then they would have 3 assessments following pass. Immediately after pass was concluded, 30 minutes, and then 60 minutes. And we evaluated response time on a modified version of the serial response time task, serial reaction time task. And what we found is that all individuals, unsurprisingly, after pass show improvements in their ability to perform the skill with repeated exposure. This is what we would expect, but there was no effect of pass on initial skill performance, or another way to look at it is what we might consider initial skill acquisition is another way to think about it. But interestingly, we had all the individuals come back a week later, and what we found is that unique to the pass conditions, both when the target muscle was active or when the target muscle was at rest, they showed a retention of the skill that had been acquired during the session. And so that's denoted here that actually not only did they retain, they actually showed an enhancement. They were actually more proficient at the retention test than they were at the end of the previous session versus the individuals who underwent a control stimulation paradigm. They actually showed forgetting. They showed that they had a decrease in their ability to perform the task. And what was intriguing is that even though we didn't see significant effects of pass at a group level, we did see that there was a strong correlation between the amount of learning, this difference from retention to from post 60 time point to retention was positively associated with how much change in MEP amplitudes we saw. So in this case, what we would interpret is that the greater increase in MEP amplitude was indicative of greater LTP-like plasticity induction. So to kind of our main conclusion here is that not all individuals respond to stimulation. This is probably no surprise to anyone who works in neuromodulation, but those who did respond neurophysiologically showed the greatest response behaviorally as well. So I'll just pause there for 1 second if there are any quick questions or clarifications. Okay. And so we also followed this up in a pilot study in a small cohort of individuals, and I won't go into detail just for the sake of time, but these are primarily from published work from a postdoc, Jackie Palmer, showing that actually, contrary to what we anticipated, we actually see that individuals post-stroke, and they're all in the chronic phase of stroke recovery, actually showed substantial LTP-like plasticity induction with a standard pass paradigm, and in this case with the target muscle at rest. But I want to draw your attention again to this variability in how much change in the MEP or how much plasticity was induced, that it's not, everyone does not show the same sort of response. The interesting thing is that contrary to young healthy adults, we didn't find any effects both in terms of skill acquisition or in retention or in learning. But what we did see, and even in the small subset of individuals, that those individuals post-stroke who had the greatest, again, LTP-like plasticity induction, as evidenced by the change in the MEP amplitude, also showed the greatest change in response times. Again, this is a very small pilot study, but it does seem to suggest that there is some association with the magnitude of plasticity induced in an individual and the effects behaviorally. Now, in sort of the last time that I have remaining before we break into the small groups, I just wanted to talk about how we've taken this idea of pass and now moved it more cortically and looking at now trying to induce plasticity in specific cortical circuits. And in this case, I won't go into all the details in terms of what's the historic interhemispheric imbalance model. There are certainly limitations to this model, but it is a model that we have used to talk about motor control and how the hemispheres communicate with one another and how that communication is disrupted to some extent after stroke. And whether the disruption is compensatory or may be facilitatory is sort of a separate conversation from this talk. But really the idea here is that we were interested to know if we could actually induce plasticity targeted in this specific circuit using paired associated stimulation. And so in this case, we use a dual TMS coil paradigm where we have a target hemisphere. In this case, in individuals with stroke, it's in the ipsilesional hemisphere is the target hemisphere. I'll refer to that as. And then we have what's called the conditioning pulse, or the pulse that precedes stimulation of the target hemisphere, which is centered over the contralesional motor cortex. And when you do this in a non-repetitive way, what you see is an inhibited MEP response when you condition on the contralesional hemisphere compared to the unconditioned response when you stimulate ipsilesional hemisphere. The thing to note is that individuals post-stroke seem to have less sort of what we call capacity in the circuit to generate this, what we call IHI or interhemispheric inhibition compared to age-matched able-bodied controls. And that even though the inner pulse interval that we elicit inhibition seems to be similar at a group level, there's a large amount of variability in which inner pulse interval generates the largest amount of inhibition for individuals post-stroke, which made us curious to look at metrics of structural integrity with the thought that those individuals who may have more or longer interpulse intervals associated with more inhibition may have more disrupted structural integrity of the pathway that this neurophysiological phenomenon depends on. And indeed, that appears to be the case, at least preliminarily, is that those individuals with more disrupted transcallosal fiber tract integrity were those who had who had less interhemispheric inhibition and whose interpulse intervals were larger that generated the maximal amount of inhibition. And just one more point on this, what's the meaning of this idea of interhemispheric inhibition capacity? We seem to observe that those individuals who have less capacity, as I mentioned in stroke at a group level, there is less IHI capacity compared to healthy controls, but those individuals with the least amount of IHI capacity were those with the greatest amount of persistent cardiac impairment. So we attempted to try and—
Sorry. Hey, Michael, can I interrupt for a second?
Yeah, please, Alex.
So in that first panel in the upper left-hand corner, why is it that the red curve is not more shifted to the right relative to the blue curve, or is it?
Yeah, so that's a great question, Alex. So it's in this particular case, so these two individuals, it so happens that this particular individual with stroke was not one of those who had substantial tract disruption, at least as a resultant in prolonged IPIs. We thought that would be the case. We expected that it was going to be flatter and shifted across individuals, but that was not the case for all individuals, as you can kind of—
I'm sorry, I didn't realize that it was just data from one individual, but that makes sense. Thanks.
Yeah. Good question. Thanks, Alex. Yeah, thanks for helping clarify that. I just want to keep an eye on the time. So good. And basically we, again, used a dual coil paradigm, but in this case, instead of assessing interhemispheric inhibition, we attempted to repetitively pair these stimuli at the interpulse interval individualized to the point on the curve, which generated the largest amount of inhibitory response for each individual post-stroke. And so that's what I'll call individualized IH-pass versus the standard IH-pass, where we took a standard interpulse interval of 8 milliseconds and delivered that to individuals. So when we look at actually individualizing based on this biomarker of IHI capacity, we see that we get significant excitability increases in the ipsilesional hemisphere and increased ability to generate interhemispheric inhibition. And this ability to generate interhemispheric inhibition was also associated, interestingly, with greater ipsilesional excitability. And I would argue that ipsilesional excitability increases or decreases post-stroke is one of the more consistent findings that we see neurophysiologically. So the ability to potentially increase excitability in the ipsilesional hemisphere may have behavioral implications. And indeed, that seems to be the case. So we also did a modified serial reaction time task where we looked at, again, the skill score on sequence movements. And we found that only really the largest effect that we saw in terms of behavioral performance was after the individualized neuromodulation paradigm rather than the standard modulation paradigm. And certainly more so than the control paradigm, where we use now a very narrow 1 millisecond interstimulus interval. In this case, if anything, either a control or inducing a sort of LTD-like effect. And even in some measures of, again, we're targeting hand muscle here. So we looked at a small battery of tasks from the Wolf Motor Function Test. And in this case, when just showing one of them, and that was checkers, and showing that you have improved performance with the individualized condition as compared to the standard condition, as well as compared to control. So I think probably in the interest of time, I will just pause here after making a few small comments. I've given you one example of sort of our maybe newer way to try and target plasticity induction in cortical circuits. And I think it presents a potential interesting model to look at how we might induce functional plasticity in other circuits of interest. And before this call started, I was also talking about getting out of, say, motor cortical circuits and looking at the interaction between non-motor and motor regions. And cortical, this idea of cortical paths may be a way for us to actually strengthen or weaken circuits that have reorganized post-stroke to try and optimize the response. And I think the other main takeaway here is that I think for neuromodulatory approaches to be effective, they need to be tailored to the individual based on meaningful biomarkers. And in this case, I gave you one example of where we're using this capacity for producing interhemispheric inhibition in transcallosal circuits as our way to try and individualize stimulation delivery. And it appears, at least preliminarily, to exert a more consistent and larger neuromodulatory effect. But again, there are all these caveats that are still there, but it's, I think, a sort of a way to move forward. And with that, I think I will pause. Is that right, Val? Or should we, what's the timing? How do we go about the next steps?
We can split in breakout rooms now, or if you want to keep talking, we still have a few more minutes.
Okay. Okay. I just didn't want to. I didn't want to cut into the time. It's I try to be a good steward of time. I'm not always the case. So okay. Well, I will now then just put in another plug. That's what the rest of this talk was, and like I said, this was time permitting. So really, all I've shown you so far is using TMS in a standalone fashion to measure and modulate cortical activity. But of course, there are limitations both in the normal adult brain, but also in the pathological brain. So for example, this individual has profound disruption of the corticospinal tract due to this stroke lesion. And for this individual, no matter how high we stimulate or how well we target the stimulation to motor cortex, we never elicit a motor response. So we learn very little about the reorganization of motor cortex in these individuals that have, again, this profound wiring disruption. And we, again, have very limited ability to characterize brain network connectivity, which if we accept the idea that behavior is underlied by networks of activation rather than siloed, discrete brain regions activating. This is a pretty important limitation to consider. So in our lab, our quote unquote solution or way we try to address these limitations of TMS in a standalone fashion is combining them with real-time imaging with EEG. So we record electroencephalography during TMS delivery, where it allows us, in this case, this is just a two-dimensional flat map of evoked responses in EEG channels, both local to the site of stimulation, but as you'll see, even distal to the site of stimulation, you do get evoked responses. And some of the work we've done, this is just kind of an example of showing this, whether we stimulate over left motor cortex versus right motor cortex, you get a very different event-related response. In this case, we call it a TEP or a TMS evoked response, whether you stimulate those. And these are just showing potential scalp maps showing the responses elicited by stimulation as it evolves across time from 30 milliseconds to around 200 milliseconds after stimulation is delivered. So hopefully you can appreciate the potential of this technique to give us more information about how networks are connected or potentially disconnected after stroke. And really our goal is to try and understand and measure cortical connectivity and looking both in time and frequency domains to basically give us a causal probe and be able to see how networks have reorganized to either support or impede the restitution of function in the stroke brain. And so we've done this in several different ways. I'll just give one example where we're looking at what are called interhemispheric interactions. So this is just looking at measures of coherence between in this case, two EEG electrodes in response to TMS. TMS is delivered at this triangle, and you can see we see sort of this oscillatory pattern between 15 and 30 hertz, so beta frequency range. And what we notice is that individuals at rest in stroke, we don't see any difference in this, what we call TMS evoked connectivity. However, if you have individuals in a active condition, in this case, during a classical transcallosal inhibition paradigm where the ipsilateral hand muscle is activated, this is where you actually see a change in the amount of evoked coherence. And in this case, what we would say the interactions are increased. We tend to think of this as they might be more tightly coupled during activity in this case. And so who cares, I guess, is the next question. Well, it seems that the more tightly coupled or the higher this TMS evoked coherence is strongly associated with the level of paretic motor impairment. So we're following up on this conceptual idea of a change in the flexibility of networks. And again, this is just one example from motor cortex, but I think the idea is to use this as a sort of a foundation into looking at, again, other cortical circuits. So this is sort of the most simple thing we could do first. And these are just some other projects that we're working on. Again, I mentioned Jackie has done a lot of work looking at these TMS evoked responses in as it relates to the recovery of lower extremity motor function post-stroke. And we're also interested in actually doing connectivity modeling where we basically input signal in one region and model its flow throughout the entire network across the period of evoked response to see what is sort of what we might consider normal flow of information versus abnormal flow, and trying to see if we can identify useful markers of that we could target potentially with other neuromodulatory paradigms. And then the last point that I wanted to make in terms of combining these, which I think is actually the future, is combining perturbation with imaging rather than using either one in isolation. I think there are outstanding opportunities to do this, especially when we're talking about precision-based neuromodulation approaches. And again, we have the advantage that we can assess multiple cortical properties. And in the case of EEG, we can actually do it on biologically relevant timescales. So we can actually see events happening in real time, unfolding in real time in the human brain, non-invasively. That we can look at, we're not dependent on an evoked peripheral response in this case from stimulation. So that allows us the opportunity to characterize evoke activity in any brain region, any cortical region in this particular case. And that you can look at different brain states. This is not, we do not require engagement of the individual during this type of stimulation and imaging paradigm. And lastly, what our lab is most interested in at this point is using EEG signatures to guide stimulation delivery. So identifying potential correlates of permissiveness of plasticity induction in real time. So taking a dynamic approach, as it were, to trying to induce plasticity where there may be a heightened receptivity of the cortical region based on, in this case, oscillatory behavior that might allow us to have more effective plasticity induction with our perturbation paradigm of choice. So I'll quit there and just summarize very briefly that I've tried to at least articulate that we think that non-invasive brain stimulation can target plasticity induction in specific circuits, that this targeted neuromodulation, I'm giving you some examples, can potentially facilitate motor learning, and that it's really important, if not critical, to account for inter-individual variability using biomarkers that we can target to optimize the parameterization and delivery of these types of non-invasive neuromodulatory approaches. And lastly, I think the future is really using these real-time integrative approaches of imaging and stimulation tools or perturbation that we can be able to not only causally perturb brain circuits, but also to causally modulate them. So with that, I will stop and again, acknowledge all the folks who made this possible, and thank you for being here, and I'll take any questions or however we proceed next.
Thank you very much for your presentation. We have 2 questions in the chat, but I think we can splitting the breakout rooms now, and we can address those questions at the end, and each group can discuss between them. So you have 15 minutes in the breakout room. A message will pop up on your screen, so please just click join when this message appears.
Well, Bella, I got a couple messages saying that the link didn't work for them. So I don't know if you maybe want to resend the link. I just saw that now, but
Yeah, I did send the link for them.
okay.
Okay, they're coming. Welcome back. Does anyone want to share with us what you discussed, or should we address the questions in the chat? So we have a few questions in the chat, and I think it's good to discuss as a group. The first question is from Ricardo Mesquita, and he asked, is the MEP size at baseline a predictor of how well the participant will respond to the plasticity protocol?
Oh, so that's a very good question. I can say that in particular in our work looking at the interhemispheric inhibition and looking at interhemispheric pass, that it turns out that that does seem to be at least the magnitude of inhibition is associated with the amount of plasticity induced. That those individuals with less IHI capacity seem to demonstrate greater response to the neuromodulation paradigm, almost in what might be considered a ceiling effect, that they actually move away from their ceiling. There is some work suggesting that perhaps baseline MEP might influence pass, but we have not seen clear evidence of that in our work. That's a really good question.
Alex Carter also asked, do these oscillations explain the variability in MEP amplitude even when the stimulus intensity and location are the same?
Yeah, so some others have done work looking at, what are predictors of MEP variability, and there has been some work showing that pre-stimulus beta does seem to, if you have an upstate or downstate, that you will see differences in the MEP amplitude. So that was one of our initial desires, was to try and reduce MEP variability by timing stimulation to see either an upstate or a downstate of that beta oscillatory waveform. Yeah, so it does.
And the last question in the chat is from Stephen Kramer. Have you found any anatomical differences that explain intersubject variability in TEP?
Oh, that's a good question. In stroke, in terms of sort of maybe like lesion characteristics or other aspects?
Well, in the simplest sense, I'm just thinking, if you induce voltage at one site and you see voltage appear at a second site, is it anatomically explained? And if so, to what extent does anatomical disruption interfere with the normal pattern of voltage spread? It could be a very clever probe of functional rearrangement, although the brain has so many connections, it's probably never that simple.
Yeah, and in fact, that was ignorance was bliss at the beginning of this. So that's kind of where we started, and what we thought we might be able to see is, yeah, we inject signal here and we just watch it move. And it turns out that it is much more complicated than simply doing that. But we do seem to have some evidence, and there's some work out of Marcello Massimini's lab in Italy showing that you can actually stimulate lesioned cortical regions, and you can get a sense of what sort of how lesioned are they by the reactivity of, elicited in other regions. So this idea, almost like some of the work that's been done in like fMRI, where you seed a lesion region and you see what the connections are. So that's one way that I know that it's been done. For us, I would say we haven't had a large enough sample to be able to say anything conclusively in terms of, if we have like cortical lesion, do we see a very clear like stop in the signal, like one would see in disorders of consciousness, where you stimulate a region and you see no propagation of the stimulus outside of the stimulated region. But that would be sort of the presumption that if you did have, lesion in the area that was stimulated, you would expect to see, it not pop up in another region.
Right. In the simplest case, and just building on the very nice models that you showed, one might wonder, I believe that from the maps you showed briefly, and they were about this big on my little screen, that there was some TEP roughly in the homologous side contralateral to the TMS site. And so whether the structural integrity of the corpus callosum, or specifically of the motor corpus callosum, is in some way proportional to the contralateral M1 TEP in healthies after stroke injures the corpus callosum or not, etc.
Yeah, and we have looked at that, and that has not been as clean as we would have liked. We have seen that they're measuring what we I guess call contralaterally evoked responses does not correlate as well with structural integrity of transcallosal pathways as I would have hypothesized or expected. But
so how's the signal getting there? I mean, that's still really cool, right? Like,
yeah, well, we think so. Especially the earliest responses, we know those are so in in the 20 to 30 millisecond TEP window, we know that's going to be transcallosally mediated. Right. Anything after that, it could be it could be several different cortical, subcortical loops that could be influencing, and that's where that's why I said it's never as simple as one thinks, because once you get outside of that time scale, now there's potentially indirect pathways that could be mediating the the signal popping up at another brain region. But we really like those earliest time points because we have some sense that if you ping the area here and at a 5 to 10 millisecond or 20 millisecond latency later you see an invoked response there, then that's a pretty strong indicator that those regions are directly connected. And of course we have some plans we'd like to do more of looking at having structural connectivity models to sort of test those ideas a bit further. Nice. Yeah, we sort of went. Sometimes you go where the money is, I suppose, and we sort of stopped doing a lot of the characterizing connectivity, and because we were fortunate enough to receive funding to modulate connectivity, and so we're having now we're coming back full circle, looking at some of these models of effective connectivity that we can, I think, build uniquely build with the perturbation imaging paradigms.
Okay, hey, Michael.
Could you please, the vanguard, man. You're at the vanguard.
Michael, could you please comment on how you are planning to, or one might plan to, stimulate non-motor regions instead of M1, right? Because you could stimulate supplementary motor regions or premotor regions or other frontal lobe areas or parietal cortex or a bunch of different things. And I understand that you might not, I mean, we don't know how that would change things like the MEP, but it seems like those regions would be potentially very important in terms of affecting both motor learning and motor performance. So can you comment on how you might do that?
Yeah, absolutely. Thanks, Alex. That's a great question. And there is some evidence that has shown, Giancarlo Caccia's group showed that if you stimulate PPC, ipsilateral PPC prior to stimulating M1, if you do that in the exact same sort of cortical paired associative paradigm, you do see modulated MEPs after that paradigm. And if you get the timing wrong, you see a reduction. So And if you get the timing right, you see an enhancement. So that actually was foundational work for us, that, oh, we should probably be individualizing the timing of stimulation, because one person's exactly right could be another person's exactly wrong if what we're trying to do. So we're really about this idea of timing is everything. But to get back to your other point, I think what ultimately, without going into too much detail, is that I think what we'd like is to have individualized network maps of post-stroke, because again, every stroke is going to be different and everyone's going to have a different connectivity profile that may either support or impede recovery, and use that to identify sort of aberrant nodes that we could target with stimulation, whether it is, so maybe for stroke survivor A, it's posterior parietal cortex. For stroke survivor B, maybe it's supplementary. Or the other approach, I would say, is that depends on the behavior that you're trying to modify. So for example, if I was really interested in visually guided reaching or motor control, I might be more interested in looking at associative areas and parietal cortex. If I was more interested in attentional, contributions to motor, I might look at prefrontal. And so it really would depend, I think, on where the deficit is. So again, always, I think, coming back to this idea of personalizing it to, the unique needs of that individual, much like we would try to do as therapists just in general, is that you're not going to treat every patient the same. Why would we deliver neuromodulation the same to everyone? And so using, again, where they are at different points in the recovery process as the guidance for where we would like to stimulate. But again, now it gives us many more tools than just saying we're going to stimulate motor cortex and just change some of the parameters. Now we can say, no, we're going to stimulate different nodes of this highly integrated network that has been reorganized post-stroke to try and facilitate the recovery of these, because in fact, I don't think we even allow it in our lab to say motor function. It has to be sensory motor function, which is maybe even still insufficient. but just getting at this idea that it's not like only motor cortex that is necessary for goal-directed volitional movement.
And so continuing with that idea of sort of like personalizing the treatment or the intervention, I think that you're right that the combination of TMS and EEG, even though I think technically challenging, is a really important way to go. Do you know if it is possible to use the EEG measurements that you get to predict the timing that you need between stimulating two cortical areas to get the type of plasticity that you want, whether it's facilitation or inhibition? Can you tell that based on the person's EEG?
I would say in theory, yes. In theory, we should be able to do that, much kind of in responding to Steve's question, that we can look, especially if we're talking about direct connections, that if we stimulate one region, we are able to see short latencies responses in other regions. So you could use that as a way to have EEG guide the timing of the two stimuli, yes. And I think maybe also a good thing to kind of come back to when you mentioned it being technically challenging to do perturbation and imaging in real time. I agree, it is a little bit challenging and a little bit maybe as an undersell, but I think it's also the opportunity to integrate imaging and stimulation offline is also potentially just as useful. For example, we're trying to develop paradigms where we're basically having a functional localizer for delivery of stimulation based on an imaging session before the stimulation, and saying, okay, this is where we see maybe an abnormal connectivity profile. We're going to then intervene at that particular locus. And so it doesn't necessarily have to be online, but that is one place that we've spent a lot of time. But I think the conceptualization is really that combining those two sources of information is going to be, I think, important, again, for this idea of, I guess, like intelligently interacting with the highly unique nervous system across individuals. So.
I was wondering, so you find the ideal parameters for one individual, but is this something that's fairly stable, or between sessions or even within a session, do you have to kind of figure it out all over again?
Yeah, so that was that was one of the recent grants we wrote, trying to say that, to look at that, and for this idea of IHI capacity. It turns out that the capacity, for example, this is just the one example that I have offhand, is that the capacity for IHI is consistent week to week. So for those individuals who have low capacity, that's consistent, at least in the chronic stage of recovery. And those who have high capacity, that's also stays consistent. However, what was less consistent, at least in our data, was the timing of when we see the maximal inhibition. So we don't have a really good explanation necessarily for that, but I guess that is one of the things that we're really interested in. Is the question is, do you have to assess this, whatever this circuit specific activity at the beginning before neuromodulation, to then tailor it not only to the person but to that point in time? And I think that's an important thing to know either way. So either way, either you don't have to do that because it is stable, or you do have to do it and then you just do it. And understanding which it is I think still one of the keys on this. And of course, if you're modulating something and you would like to have a persistent effect over time, well, yes, we would like it to change, and so that could be used as an outcome measure rather than saying, oh, this is unreliable. It's actually, no, you've induced now repetitive inductions has induced a persistent change in whatever that marker is, which is of course what you would, I think, presumably want, given that it was the marker that you're trying to intervene with anyway. Yeah. Good question. Thanks.
Does anyone have more questions? So thank you very much, Michael, for sharing your time and your results of the studies that you have been doing with us. I hope to see you again on March 11th. The speaker will be John Krakauer from Johns Hopkins University, and we will discuss the topic, thinking about motor recovery after stroke. So thank you very much, everyone.
Well, thanks everyone for being here, and thanks for all the questions. I appreciate it. Yeah, take care, and feel free to reach out if there are any other questions. More than happy to talk more. So thanks.
Thanks, Michael.
Bye.
Great to see you. Appreciate it.
Yeah, good to see you, Alex. Yeah, thanks.
All right, bye-bye.